Thank you for coming. So I'm going to start by talking about DNA match statistics and what they mean and what they are. And then I'll go through a long case example of a case we had in California, going through admissibility, the trial, the outcome, and so on. And then I'll talk more about the use of True Allele in New York State, as well as in the rest of the country. Pete asked me to take under an hour to go through that, and then we'll have a question and answer, because I'm sure if you're still awake, there'll be questions. So I'm calling this talk No DNA Left Behind, because that's the goal. We don't want to leave DNA evidence behind. And the subtitle is When Inconclusive Really Means Informative. And I'm sure that in the last 2 years or so, you've been seeing more DNA reports that say the answers are inconclusive. I'm going to introduce this by explaining why that's happened. So the first thing is a DNA match statistic is a key part of DNA evidence, right? If the match statistic is 2, it's less useful than if it's a quadrillion. If it's really 1,000, that's really what it is. And match statistics can even go the other way. If they're 1 in a million, they can be exculpatory. So match statistics can do all sorts of things. When scientists talk about match statistics, we don't, at some point we stop talking about quintillions and quadrillions, and we look at the number of zeros. And that's the standard measure of information in science, not just forensic science. So 1,000 has 3 zeros in it, a million has 6 zeros in it, billion has 9 zeros, trillion has 12 zeros, and so on. So on many of the slides you'll see, we're counting the number of zeros. We're not labeling it billion or so. You'll see the number 9, and that means 9 zeros. So I'll be translating, but that's what a lot of these graphs mean. Okay. So this is a study that we did with Virginia, who's actually going live this week with True Allele in their own state. And it was a study where they had sent us over 100 cases, and these are all mixtures, typically with 3 contributors, some with 4, some with 2. And what we did is we looked at the genotypes. You'll learn what a genotype is in about 2 minutes, but it's a genetic type. And we looked at the match information. And so when we use the computer, which is designed to assess information, this is what we saw. On a scale of 0 means no zeros after the 1, that's just 1. A number like 10 is 10 zeros, which is 10 billion, right? It's 10 with a billion. 20, I guess, is twice that, right? The numbers get bigger and bigger. When you get out to a number beyond 20, that's basically a single source profile. So the true range of information that you see from DNA mixtures spans the entire spectrum. And this is when there's a positive association. I'll show you negative results later on. You can get a match statistic around 100. You can go out to trillion, and you get everything in between. That's what you expect. If you don't know anything about the composition of a mixture, there's a complete range of information. Human review, which is called Combined Probability of Inclusion, or CPI, which is what you are used to seeing in your reports. On the same data, virtually all the same, this is about 100 genotypes that were used, this is about 80. Instead of an average match statistic being around 100 billion, which is 11 zeros, that's what this 11 is. As a match statistic, CPI, which is the usual mixture statistic that you're used to, concentrates the match statistic to around 6 zeros. It's in the millions. There are reasons why it does that, but that's a different talk. I'm not going to go into that today. It's on our website. I also gave that at the American Academy meeting last year, as I did with this talk. Now, this is how people interpreted mixtures by eye using manual methods until 2010. Then in 2010, the Scientific Working Group on DNA Analysis Methods, the acronym is SWGDAM, came up with a policy that changed the match statistics. And I'll talk about why in about 10 minutes. And the result of that change, as we measured in the study with Virginia, was that the average match statistic was 6 zeros or a million, and then it shifted to 2 zeros or about 100. And that's when there was a result. In the study, maybe half of the DNA was interpreted, and a result was obtained. That doesn't mean they couldn't do it. It just means in the study they didn't do it. Sometimes you can't. The others would be inconclusive. So question is, why are you getting less information with human review than with TrueAllele computer review? And why are you getting far more inconclusives? And when you do get a match statistic, the average is now in the hundreds instead of in the millions, which is what you were used to 3 years ago. Okay, so that'll be some of what I'm addressing. But this is kind of a mini national crisis for prosecutors, because evidence that was informative 3 years ago, today the same evidence is uninformative. And that's what happened with Virginia and why they contacted us. There were 375 cases, the guidelines changed, and suddenly the reports had to be retracted. They couldn't report on what they would have reported before. Okay, so I'm going to begin by describing what a genotype is. Most of these slides we use with juries, so there's nothing here that's terribly technical, and if you have questions, we can discuss them afterwards. The key concept is the genotype. That's what scientists are looking for in forensic science. You have your chromosomes, right? So you have a person, they have about trillions of cells with nuclei in them. Each nucleus has 23 pairs of chromosomes. That's basically packaged text. They're like volumes in an encyclopedia, and the DNA machinery reads it like one-dimensional text from a book. They're instructions. If you open up that text, you can find regions that are locations that are called, one is a locus, many are called loci. And when you zoom into an area that has maybe 100 to 400 DNA letters, the DNA alphabet's written in 4 letters, A, C, G, and T, that correspond to 4 different molecules. You can read out certain words or sentences. A typical sentence, but you can think of it like a paragraph or a sentence in a book, maybe it's 200 letters long, and for identification purposes, in the center of it, there is a repeated word. Those are the DNA regions or loci that are used in forensic science. And the reason is there's a lot of variability. If you used a location that had no variability, like maybe a gene for blue eyes or brown eyes, you wouldn't be able to distinguish people. If you use a region where there can be 10 or 20 different values called alleles, and then you have a pair of them, one inherited from your mother, one from your father, then you have about 100 different possible values, a 517 or a 1012 or an 88. And these numbers correspond to the lengths of the molecule. And the lengths are determined by repeated units. There are about 100,000 of these believed to be junk DNA regions throughout the human genome across the 8 billion letters. And what they have, as you can see, is you have a starting point, and then you get to a repeat region. And so some 4 or 5 letter word will repeat maybe 10 times, and then it goes on again. And scientists capture that entire sentence, including its repeat region. And if one sentence has a word repeated 10 times, and maybe from another parent you've inherited a sentence where the word is repeated 12 times, there'll be a difference in the length of the sentence. One will be longer than the other because it has more of that repeated word. And that's the basic of the genetic variation that's used in forensic science. And an allele is one of those repeats, segments, like with length 10 or length 12, and a genotype of an individual at a locus is the pair. It's the 1012. So one person may have a 1012, another person may have an 88, whatever the pair is, that's their genotype. And that's what scientists are trying to work out. Okay, so the DNA identification pathway is fairly straightforward, and I'm going to show it to you for simple DNA, where you only have one contributor, and life is pretty easy. So you have an evidence item. It contains one individual. The DNA is extracted by the laboratory, and then it's amplified, and then experiments are done to put labels on these amplified fragments and read them out based on size. So 10 repeats has a smaller length than 12 repeats. The heights correspond to quantity. But with one person and abundant DNA, if you see in the data a peak at 10 and a peak at 12, that's a very easy genotype. Of the 100 possibilities or so, there's really only one genotype, and you can see it from across the room. There's only one possibility. The person has a 10-12 genotype. That genotype can then be compared with a known genotype, and if there's a match, you can produce a match statistic. Okay, so this is the simple case we're moving away from very quickly. What match information tells us is the change in information from what you would have known about evidence before you did the test to what you know about evidence after you do the test. It's a measure of change. So if you begin with what's the probability of a coincidental match in the population, the chance of seeing someone with a 10-12 with this genetic locus might be 5%. Okay? So there are 50 or 100 genotypes. Maybe this one is more prevalent. Its chance, if you took a random person, it would be about 5%. Well, after you've looked at the evidence and you've seen that it's a 10-12 and it matches the suspect, the chance of a match is 100%. The evidence is a 10-12, the suspect's a 10-12, there's complete certainty it's a match. So what's the change in information? You went from 5% before you saw the data to 100% chance of a match after you saw the data. 100 over 5 is 20. At that location, the match statistic is 20. If you have many locations, you multiply these numbers together, which is how you get very big numbers. If you multiply 10 times 10 times 10, 15 times, you get 15 zeros. Okay? So that's what a match statistic really means. It's just the change in information from before to after, and you can calculate it. Now, what happens with mixtures is you no longer have only one or two peaks corresponding to one or two alleles, like a 10-10 or a 10-12, because there's more than one person in there. Okay? That's why it's called a mixture. And a mixture will produce quantitative data. Suppose that there's one individual who's a 10-12, which you see as two lower peaks, and another individual who's a 7-14, which you're seeing as two taller peaks. This is what the data looks like. On the x-axis, you're looking at a measure of size, how long the DNA sentence is, and then from that, using other calibrations, scientists can determine that the length of the sentence corresponds to 7 or 10 or some number of repeated units inside the sentence. And the height on the y-axis these are measures of fluorescence that tell you a measure of how much DNA is present. So what you're seeing here is that there's one individual whose genotype is a 10-12, and another individual whose genotype is a 7-14 with about twice as much DNA. So you're adding together a small amount from one genotype and a larger amount from another genotype, and that's the best explanation for a pattern like this. Okay, so that's what a mixture looks like, and notice there's a lot of information here. You know what the size is, which tells you the allele. You also have information about the height, measured as a number. That turns out to be very useful information. Now the problem is that the simple DNA pathway that you have of saying, well, the lab is going to take this mixture of two people, the lab will produce evidence. What human review doesn't do now is it doesn't produce a genotype anymore. It's not saying, here's the genotype of one individual, here's the genotype of a second individual. It does other things, which you'll hear about. And since you don't have a genotype, because there's some uncertainty in the data as to what the genotype would be, the simple approach of having only one contributor doesn't work, and you don't get a robust way of getting a match statistic when you make a comparison with a known individual. I mean, people do things, I'll show you what they do, but it's not a great thing. It's not, it throws out most of the information. Okay, so the human interpretation issues are these. A lot of good data is called inconclusive. When the peak heights are too low, they're not used. When there are too many contributors, like 3 or more, the methods don't work very well. And there's also potential examination bias in the sense that the match statistics are designed to say that when, here's one example, suppose there are 15 different loci. If you see that an individual is included at 10 of those loci, a match statistic will be reported at those 10 loci. What about the other 5 loci? Well, they're not used. They may have been more exculpatory than favoring the prosecution, but the numbers aren't used, and that can lead to interesting and often valid arguments by defense experts who say, well, you did it one way, I did it another way. Isn't this subjective? And there are a lot of articles that have been written about that for mixture interpretation in the last few years. I won't go too much into DNA databases because I'd rather talk about New York State, but the problems of what happens when you don't have genotypes, but you just have the numbers, the alleles, 7, 10, 12, and so on, extend to databases, and they make them less powerful than they can be. CODIS is a fantastic tool, but it doesn't use all the information in the data. And so much mixture evidence, if not most of it, is never uploaded to CODIS. And there are reasons for this. The math is not used when comparisons are made between evidence and suspects in the CODIS database. Because of the nature of the comparisons, there can be false hits. In order to compensate for that, the CODIS policy is to restrict what can be uploaded to avoid the false hits. The result is you end up losing true hits, and though it works very well in some cases, the information is lost in others. So the system I'll be talking about, which New York State Police have, is TrueAllele Casework. And there, on the evidence side, the whole point of the system, which we started building 15 years ago, is to preserve all the DNA information. Everything that's in the data, we're trying to preserve all that identification information. We use all the peaks, whether they're high or low, any number of contributors, and the method is completely objective in the sense that when a genotype is inferred, the system has no knowledge of the suspect or victim you're making a comparison to. So it can't be biased by what you're comparing because it doesn't know. And at the end, we report on all the loci, whether they're favorable or not. There's also a database, which I think New York will have at some point, which again uses genotypes and all the information that they have. It's based on match statistics, not just association. It's sensitive in that studies that we and others have done show that it finds true hits, and it's specific in that it doesn't find false hits. Therefore, you don't need to restrict the upload. Everything can be uploaded, and you can make full use of all the identification information. Okay, so how do you restore the data pathway? The concept is to use science where uncertainty is represented with probability. You can't just put down any old numbers. You actually need a mathematical model and computers to do this. But once you do, you can represent an evidence genotype out of the 100 possible pairs of values. You're not going to get just one because it's not simple data, but you can restrict the possibilities to allele pairs like 10-10, 10-12, 10-14, and put numbers next to them that add up to 1 or 100%. And so, like in the rest of science, you represent uncertainty with probability, and then you move forward. And it turns out you can compare a genotype that has probability with a known genotype from a suspect or a victim, in cases where you have mixture and more complex data, and produce a match statistic. And this is how it's done. If the probability of the suspect's genotype is, say, 50% in the evidence. It's no longer 100%. It's some other number. It could be 2%, 30%, 50%. But it's now some lower number. Relative to a coincidence, instead of getting 100 over 5, you get some lower number, like 50% over 5. And now you can produce a valid match statistic. The value here, say, is 10, because it's 50 over 5. And you now have 15 or so different locations, different loci. You multiply them together, and all the statistics work. So computing a statistic is no longer hard. The hard part is actually computing the genotype. That's where a computer can take hours or days, depending on how hard the problem is, to solve for what these genotypes are. But it's the same concept. You start with no information before you look at data. You get information afterwards. And you ask, at the suspect's genotype, to what extent do we have an identification? That's in the numerator. Relative to what extent we have coincidence? That's the denominator. And that's how match statistics work. So now I'm going to talk about a case. This was in Kern County, California. Kern County is a farming and mining community about 2 hours northeast of Los Angeles. This is not a true map of Kern County, but this is the case. And what happened is that Kern is on an interesting path between Mexico and Northern California. So they got a lot of people coming in and out. And they have a problem with gangs. And this was a gang that came into the county, and they began robbing stores. They robbed a food mart where it was a very gentle attack, and they left a gun and a hat that could be analyzed for DNA evidence. They analyzed other stores, other locations, a hardware store, and as the gang got more experience, like with not letting victims run away and so on, they got more violent. And the crimes became more serious, and they kept leaving little bits of DNA evidence. Now, the issues with this DNA evidence is most of these were low-level 3-person mixtures. So the lab looked at this and said, this is not something that we're allowed under the current guidelines to produce a match statistic on. But the lab at the same time had recently purchased our TrueAllele system. In fact, they were the first group to go live with it in the US last fall. And I've helped them with several cases there. But it wasn't on board yet, so I was more involved. Now, in addition to multiple crime scenes with different items of evidence, so 12 evidence items across 5 crime scenes, they also had reference items, 5 victims and 5 suspects. And the question was, how do you match up the genotypes from the evidence items to the genotypes of the suspects? That's the question. And if you can match them up, what sort of statistics are you getting? So the goal is not just to get match statistics, it's to determine who is at which crime scene and who is doing what, who is handling which evidence. So there's a lot of information in the DNA. The way it worked is that at Cybergenetics, we received the evidence data from the lab, but no references. We started the computer processing. We always replicate the results. This is because the equations are complex. They involve maybe 100 variables, and they're all random probability equations. The way you solve these equations is using a random search procedure, and if you get pretty much the same match statistic after you run it twice, that means you're getting concordant answers. You can read a lot about that, but I won't go into that today. If you have questions about the math at the end, I'll be shocked, but I can answer them in what we've written. Later on, we received the known references. This was an interesting test because it meant everything was completely objective. We know that our process has no idea what it's going to be compared against, but in this case, we didn't know. We then made the comparisons, calculated statistics, and on the 12th day, we sent the match results back to the lab. This particular lab is part of the district attorney's office. And so it works very well in that county, and I've worked with prosecutors there. And their goal of doing very accurate investigations and getting all the information they can, whether to decide whether to prosecute or to use it, is working well with their DNA lab. I don't think that's the usual case in the U.S. Okay, and then this is what we found. The computer didn't draw this picture. I drew this picture for a conference talk. It's in a paper that I gave at Promega maybe 2 years ago. Yes, the last Promega conference paper I wrote. And I gave a talk at that meeting. And then this picture is more informative than just showing you a bunch of numbers or match statements. What it's connecting is for the different items of evidence shown in different colors at different crime scenes, we're connecting them with the 5 suspects. And each edge is, for example, this green edge connects the countertop in a jewelry store to suspect number 2. And this information is in tables that we provide, as well as in match statements and reports. But you can see how you begin to figure out, individual S5, they have doing a lot of work at these different crime scenes. Other people are doing less work in a hands-on way. So the match statistic of all these matches that were found that was of interest to the first trial was a match statistic of around 500 million, and that was in the people of California versus Lawton and Langston. The trial started in November of 2012, and I testified in January of last year. They have long trials in California. And in this case, it was the first use of TrueAllele in California, or in Kern County, and there was an admissibility hearing and trial testimony, which is what I'm going to walk through. In the top left corner, I have a little icon here with a gavel that's indicating we're at a Kelly Fry hearing. Okay, it'll change as we talk about different things. Something that's very good to have is peer-reviewed publications that are validation studies that establish to what extent is a method sensitive and that it finds correct answers. Is it specific? It's not implicating the wrong person. It doesn't get wrong answers. And reproducible. If you run the computer twice, or you have maybe 2 people manually solving a problem, are you getting the same answer? So we've done a number of validation studies, probably done about 20 validation studies with different groups, but we don't write everything up. We maybe once a year will take an interesting study. And a peer review is a arduous process that can take a year or 2. You write up. Are you familiar with peer review? Scientists will take their results, write a paper, and then send it to a journal, who will then send it to 2 reviewers, and then they have complaints or criticisms, comments, and then you address them, then you send it back. And then they have more comments. And it's kind of like court. You just go back and forth for a while, and usually it converges, and after all the concerns have been addressed by your critics, then a paper's published. So it's sort of a scientific approach of having a system with a judge who's the journal editor and 2 parties coming back and forth. So here are 4 validation papers. 2 of them, shown in dark blue, were done with the New York State lab in Albany. They were both published in the Journal of Forensic Sciences. The second one down is called Validating TrueAllele DNA Mixture Interpretation. I'm the first author, and Dr. Barry Duesman, who's the Director of Biological Sciences at the Forensic Investigation Center in Albany, is the last author. And then in November, at the bottom, you see we published another paper with myself, Jamie Belrose, who is a graduate student now at SUNY Albany, and with Dr. Duesman, was a much more extensive study on the New York State TrueAllele Casework validation study. These papers describe the methods, results, case examples, and then a lot of statistics and charts describing, not proving that something is reliable, but demonstrating the extent of its reliability. reliability. I mean, it's science, so the goal is to show how far you've gotten. So validation papers are very helpful. This is an interesting slide that shows relative to how much of an individual is in the mixture, because these are all mixtures, how many zeros are in the match statistic. This data was done with the Kern County lab. We're writing up other papers about our work. And each point is telling you the fraction of that genotype in a mixture and how big the match statistic was. You get a whole bunch of points, you get a scatter plot. Okay, this is, other groups do this as well, and it shows generally that the more that a genotype is present in a mixture, on average, the larger the match statistic. There's a line that's going from left to right and going up. The defendant's point for the reported match is right in the middle of that cluster, which indicates there's nothing unusual. And that's often nice in a admissibility hearing to show that the case in question just falls into what the validation studies show. This, I'm now going to show you match statistics that go the other way. Let me show you here. A -6 is 1 over a million. It's a million is a 1 followed by 6 zeros. -6 means 1 over that number. A -12 is 1 over a trillion. A -18 is 1 over a million. So what this is showing is how specific the system is when you take computed genotypes with probabilities and compare them against other individuals that are not supposed to be in that case. They can be randomly generated, they can be from different cases, but you're showing what's the degree of exclusion. And that's what these red bars are all about. That's the distribution from -20 up to maybe -6. So that's 1 in a million, and that's maybe 1 in a million. This is the distribution of how often you see these exclusions. And notice in this particular study, we're not seeing anything past 0, which is a match statistic of 1, which is completely inconclusive. And there's the defendant. So visually, this shows what the match statistic is trying to tell you, that if you compare against thousands of individuals who are not the defendant, then he's completely far away. He's out of a billion, and there's nobody even close. Okay? So that, again, is showing something about the reliability of the method in a particular case. Okay, so now I'm going to move on the icon change to a jury. It's supposed to be a jury, up in the top left, if you can see them. And the idea, again, is computers can use all the data. They use the peak heights. They look at the entire pattern. So there's a pattern of peak heights. There's variation that can be modeled. There's just a lot of information in each peak as well as in the pattern of peaks. And the computer uses all of that. But people don't. It's a very hard mathematical problem, and what people do is they simplify. They do what they would have done if they had a lot of DNA from one person. They draw a line, and they say, what are the 1 or 2 peaks that I see that would correspond to the allele? And that works great for a reference sample, but it doesn't work on mixture evidence. When there's a minor contributor, or there's more than 2 contributors, or there's low levels of DNA, it's getting the math wrong. It's an approximation. It's often close, but it doesn't work that well. And so what happens is they draw a line, and then anything over the line is said to be in. Anything under the line is said to be out. It's not used. There are some other variations on these methods with 2 thresholds and things that you can do for inclusions and exclusions, but look at what's happening. You're taking the pattern of data and you're reducing it to a bunch of events, like numbers, saying that entire pattern is now 1415. it's those two events that are in. You don't know if they're really in. You don't even know if these events are really out. They look like usable data, but you're not using them. So now the pattern is gone. The quantity is gone. You're not saying how tall those peaks are. And the other thing that is more for a computer is the way you use the pattern to determine what the variation is around those peaks. If you were to repeat the experiment, you'd get a slightly different pattern. Computers can figure that out from the data. So you're losing the pattern, the quantities, and the variation, all of which you need to get all the information out of the data. So this is what the computer does instead. The computer sees the data, and it tries out tens of thousands of different patterns. It puts possible genotypes where the data are, where the data aren't. It puts them in different heights, different proportions, different amounts. It also worries about many things that you're not seeing on this picture, which is the degree of artifacts, like stutter and peak imbalance, just things that happened during the laboratory experiment. And what it does, so here's a pattern where there's a lot of a first person shown as a pair of blue peaks at alleles 14 and 15. And so there's a pattern where it says, suppose that's a major contributor. And then in orange, there's another person's allele pair at 12, 13. And then in green, there's another person's allele pair, but much lower, at 10 and 16. That pattern explains the data very well. Therefore, that has a higher likelihood. It's more probable, and that ends up, through the math, conferring more probability to those three genotypes, those allele pairs, for each of those three contributors. The computer tries out a lot of other patterns, too, most of which have much lower probability, and most of what it tries out for the 100 possible genotype values for each contributor ends up with essentially no probability. So that's what the computer's doing, is it's generating possibilities, comparing them against the data, and ask what better explains the data. Importantly, not what best explains the data, because then you would get a probability distribution. You'd either be right or wrong, and now you're almost back to where you started. What you have to do is a computer search where you're trying out virtually everything and asking, by how much is each thing better than something else? So when you're done, you end up with a probability distribution. And that's what a probability distribution looks like. What you're seeing are all these different possible allele pairs. There's 100 allele pairs would go from the left wall over to the right wall. Those aren't even on the picture. So imagine that when you started, there was all this low-level probability of a random person. Now the computers looked at the data, and at this one genetic locus, for this one of the three contributors, it's concentrated the probability where the data has told it to concentrate probability. It only knows what the data is showing it. And it's put most of the probability on 14, 15, a lot of probability on 12, 13, and some probability in other locations. So you see there's a rise in probability some places, a fall elsewhere. Okay, that is an objective genotype that the data is trying to tell us, if you can analyze it with math and computers, of what is the genotype. We don't know what the answer is. We only know what the data can tell us. We now get to the key point where this is going to hit the courts. There's going to be a match statistic. So at this one locus, how do we compute a match statistic? And the question is, how much more does the suspect match the evidence than a random person? And this is how we answer it. There, in these faint brown bars, is the random person. Right? That's the chance of a random person from, say, some ethnic group having those particular genotype values, those pairs of alleles. In blue, we see the same picture of what the evidence is showing. We still don't know who the suspect is, but now, for the first time, we say, show us the suspect's genotype. If you pick the genotype, like 1214 over here, you'd see a loss in probability, and that would be more exclusionary. At other genotypes here and here, the blue bar is taller than the brown bar. That's a gain in probability. Well, what's the genotype of the suspect? Now we tell the computer. That genotype is a 14-15, so we move this little red cursor to show you to focus attention right there. And the math of match statistics says that to a first approximation, virtually all of the information of the match statistic is just looking at that one place. And so we look at that place and we see the blue bar is taller than the brown bar. That is, the evidence genotype is taller than a coincidental genotype for the suspect. That means it's going to be giving a statistic that's bigger than 1 because it's the ratio of the blue bar to the brown bar, which in this case is about 51% over 6%. And so that's an 8-fold increase. That factor increase you can visually see as the match statistic. Why? I don't know. You can read all the math in the appendices of these papers, so you can take everybody's word for it, but at least you can see it. It's not some number that somebody's pulling out of a hat. It can be visually evident. So there's a number, a little bit less than 8 when you account for relatedness between people and so on. There are adjustments that are made. Those are the 15 match statistics. Here's the amount of the statistic. It's technically called a likelihood ratio. All the match statistics that are used at some form are likelihood ratios, just some use more of the data than others. And those are the 15 genetic loci. Those numbers are multiplied together with the result that you get a number like 500 million. And so to answer the question for the jury, is the suspect in the evidence, we can state in a report and a trial that a match between, in this case, the front counter and defendant Dupree Langston is 553 million times more probable than a coincidence. And that's language. There are many ways to state a match statistic. I and others do research on this. There's different mathematical formulas. They all give you the same number, but some of the ways of writing them down have words that are more like English, like probable, match, and so on. I won't bother you with some of the ways of stating this that are incomprehensible. You can read about them. You can ask me about them afterwards if you're really interested in how to incomprehensibly state a match statistic. But you might have experience with that anyway. Don't know. So now I'm going to show a nice description of what happened in this case. This Kern County News Channel did several segments on TrueAllele, at least 2 on this case. I'm not involved in it, but I'll play it for you, and you can see, I think this came out in February of last year.
New at 5:30, prosecutors are hailing new DNA technology that helped secure 2 convictions in a string of recent robberies.
The new True Allele Casework system made its debut in a Kern County courtroom this month. 17's Kiyoshi Tomono shows you how it works.
Charles Lawton and Dupree Langston might have gotten away with robbing this check advance store if they hadn't done this. Planting a bare hand on this countertop to vault over. In the fraction of a second Langston's sweaty palm touched the counter, it transferred a minute amount of DNA that would lead to his conviction.
And so then the technicians went back to that very spot and did some swabs.
Before, the crime lab might have struggled to do anything with evidence like this. Only a trace amount of DNA and contamination from other people who touched the same counter.
And that used to be the biggest problem, was when you had a mixture of profiles. And so you couldn't pull out exactly who was in there, who wasn't in there.
So in those cases, we would have to say that the DNA profiles were inconclusive in our analysis. With TrueAllele, we don't have to say that any longer.
TrueAllele is high-powered computer software that uses mathematical formulas to sort through contaminated evidence.
This is the first use of TrueAllele west of the Mississippi, really.
It can then pinpoint with a high level of confidence whether a particular person's DNA was present at the scene of a crime, even if they barely brushed an item.
We're talking about touch DNA. We're talking about DNA with that's been out in the field for a while and may be degraded. We're talking about missing persons cases potentially.
It is a powerful tool with a caveat.
DNA, it is time consuming and it is expensive. And so we can't do it for every case that we would like to.
In all, Lawton and Langston were convicted of 8 robberies in a 5-week crime spree, including a holdup at this Max Muscle store in 2011, where the owner shot back. Other victims said they were brutalized. Some were even forced to disrobe at gunpoint. Now that the technology has been proven in court, the crime lab says expect to see more of it.
With newer technologies, we're able to give more definitive answers.
In downtown Bakersfield, Kiyoshi Tomono, 17 News.
And a footnote here, Langston and Lawton are set to be sentenced on April 5th. They each face about 70 years in state prison. The new DNA software, by the way, was paid for with a $100,000 grant from the National Institute of Justice.
Okay. So they actually did get sentences that I think each exceeded 70 years. So I'd like to now quickly tell you, you'll recognize that icon. So these are things that have happened in New York. The state lab began working with Cybergenetics about 12 years ago. They first brought on a system that we had developed for the British government that was used to build their database that was only for reference samples, not complex mixtures. And New York State brought it online, and so they brought it online where the arrow is in summer of 2007, and this was the growing backlog that they had, and then it went away. They were able to move much of their staff into casework and reduce the voluminous documentation that they had on each genetic analyzer run down to a few pages. It was all automated by computer. We also reanalyzed the World Trade Center data for New York, reanalyzing the 18,000 victim remains and producing genotypes, putting that in a database and comparing it with 2,700 missing people, both from personal effects and from family kinship data and associating them. This comes from the first paper that we published in 2011 with Barry Duesman at the New York State Police in Albany. We had looked at 16 cases. They're mainly sexual assaults. Half of them, the victim was known. Half the victim wasn't used in the analysis. These are where the victim's not used. And each of these clusters, these 8 clusters, shows a different case item, a different mixture. Blue is showing the information results. This is the same scale counting the number of zeros of information. Again, 10 would be 10 billion, 15 would be a quadrillion. It's the number of zeros in the match statistic. So blue is the computer's results, and orange was the reported human review result. And what you can see is there's a difference between the two, and the difference is about 6 zeros, or a factor of 1,000,000. So these are cases where New York reported a result. We did another study on more items, and this was reported in the 2013 paper with New York State, where the issue wasn't whether or not New York had reported a result. We just looked at a lot of evidence items. Here again, you see this axis of the amount of information, 15 being a quadrillion, with 15 zeros after the 1. The blue background, sorted in descending order, shows the amount of information across these 85 different genotypes, about 81 mixture items. Some items had more than one genotype and more than one match. So these are the match statistics that the computer found as that blue curve. The average was around 15, zeros were around a quadrillion. These colored bars are 4 different methods that are done with human review and thresholds. The one in gray is a method where you assume that the major contributor is so large, maybe 90%, you don't treat it like a mixture. There you get the same answer. The green method is one where the victim is known, and so they can get more information. And the orange is the most prevalent method where you don't make any assumptions about the contributors. These are all largely 2- and 3-person mixtures. So you see two things in this picture. The first is the amount of information. The difference between the orange bar and the blue bar is that same loss of information that you saw. The match statistics are lower. But what's even more visually apparent is that of the 81 or so genotypes, only 30% of them have a human score. That is, most mixture information isn't being reported. There are reasons for it. Sometimes it's the case that they have a result. But think about what that means. It's like Russian roulette. You just keep firing DNA evidence until you get something that's easy enough for a person to interpret, which is a lot more work and time than taking your data, putting it into a computer, and most often getting an answer. So that's basically the thing about that is that it's showing that most of the time people are getting less information or no information from highly informative data. New York State went on about a 2-year journey down to Manhattan to the DNA subcommittee of the New York State Commission on Forensic Science. It's kind of like a peer review process. You keep coming back and showing them your validation studies. They look at it and they say, okay, well, we want more. And finally, at some point, they stop and they say there's enough. So that was approved in May of 2011, and the next month, their binding recommendation was approved by the Commission. So New York State has approval for using TrueAllele Casework in their laboratory. So far in New York State, while we're waiting for New York to come on board, we've been sent cases in 6 counties: Cayuga, Chemung, Schenectady, St. Lawrence, Tompkins, and Westchester. They're all murders and rapes. That's usually what we get. If you're going to bother to have DNA reanalyzed, it's typically for a serious crime where you have something that looks like informative DNA, but nobody's giving you a match statistic or will testify about it. TrueAllele has been used in criminal cases. We've written about 150 case reports. This number is going to go up, I think, quite a bit. Virginia's come on board with it, with their first case reports. The Kern County group is now writing their case reports, and New York State's supposed to be going live with this. I'll mention that in a second. So it won't just be Cybergenetics. Actually, there are foreign groups overseas that are also using TrueAllele. We've given testimony in state, federal, military, and foreign jurisdictions for all sorts of crimes, but they're usually serious and violent. TrueAllele, once the New York State Police are running it, I expect will be used to solve a lot of property crimes and make connections between them. The one group is running a database, and they started it earlier this month, and the first week they found a link between a property crime and a sexual assault. The next week they found one between 2 property crimes. These are connections that the usual human review and using CODIS are not finding. There have been 3 in green, so this is the system is spreading out. Different states are getting services, but in terms of admissibility, there have been 3 admissibility hearings in the U.S. There was one in Pennsylvania back in 2009. There's an appellate ruling on that and a state Supreme Court ruling on that affirmed the use of TrueAllele in Pennsylvania. And last year, we saw admissibility hearings in California and in Virginia. There'll probably be more in California. They have a lot of admissibility hearings in California. So I'm wrapping up now. In case you're wondering when you could ask questions, walk out the door, or get donuts. So the point is, we do have a computer age now, and there are other groups developing similar systems. in their first stages. We started on this system 15 years ago, and we went through 25 versions of it until about 10 years ago on our test data. The system was doing nothing stupid. And in my view, it takes that long to build a robust system. But there are others that are also being developed, so it's becoming more of a trend. The point of them is they can be objective, reliable, and truth-seeking. I mean, you can read this, but they solve the DNA mixture problem because it's not really a problem if you're using validated math. With low copy and degraded DNA, like in the World Trade Center, the math doesn't care. The computer will produce genotypes. We have different measures of what accurate means, but the numbers are reproducible, and they are also based on some measures on sets of information, like the first picture I showed you where it filled spanned the spectrum. You can use that to support the concept that the numbers are accurate. It's automatic. Instead of labs having analysts having sometimes an enjoyable 2 days looking at unsolvable data, and then when they're done they say it's inconclusive, it goes into the machine and those people are doing something more useful because the computer is working on it. Yes, it's backlogs and costs, finding criminals. It's also used for exonerations. And the major goal of what we're doing, as you're going to hear in 1 minute, is to create a safer society, and that's really what's driving us. There's a lot of information that you can get. Actually, if you'd like, I can send you the slides as a handout if you want to send them out to people, so people don't have to write all this down. We have the different papers and precedents, book chapters, magazine articles. There's just you can attest to, with the 3-inch binder that you took home for your Christmas or New Year's reading, double-sided. And that was a small subset. There's a lot you can be reading if you want to have more information. And then we have about 50 videos. This will end up as one that are narrated. You see PowerPoint slides and hear somebody talking, often me. Presentations, and they include validation studies, explanations of the technology, CLEs, just different lectures that we give at scientific conferences and legal presentations. Okay, so this is my last slide. And the point of this is really the concept of no DNA left behind. There's a concept in society that when you have a problem, like in medicine, that everything that can be done is being done. And certainly practitioners in DNA evidence know that's not the case. And that I'm just informing you, and you can read a lot. It's in the scientific literature with lots of different groups writing articles about this. But much more can be done with the data. And if you have a case that needs DNA evidence, I think you want to be doing all that you can be doing. The TrueAllele Casework at the New York State Police is installed. It's been validated. They've written up their internal validations that they've done independently. We've trained about 30 analysts at the lab, and they've been, I think 25 of them have certification as operators who can run the technology, and they've documented their protocols. They're pretty much ready to go, and the last I heard is they should be online, though one never knows with government, possibly in March. That's the latest that I've heard. However, you may have cases before then. And so I'm going to say this word that is amazing in government, free. New York State already bought this system, okay? I would like them to use it. I'd like you to know about it so you can ask them to use it. Now, how do you know what to ask them about? Well, if you just start asking them to use the system, well, hopefully in March going forward, there'll be cases and they'll be starting to process a lot of the mixtures. What if you have current cases? What if you have something where the case hasn't been brought to trial and there's important evidence? How will you know if it's important? How do you know whether to bother them and ask them what to do? send it to me. We'll screen it for you, no charge, and we'll tell you if there's information there. And then you can contact the lab and say, using the same system that you have, we know there's a lot of information here that could make a difference in our case, and it's important. Could you please run it, write a report at no cost to you, and then send somebody to testify about it? Because it's the state lab. It's no cost. So if you can think of cases where DNA might be important, and you can wait for the lab to do it, we're happy to screen it, tell you what the information is, so that when you contact either the Director of Biological Science, Dr. Barry Duesman, or the Head of Laboratory Services, the new Director, Mr. Ray Wickenheiser, over in Albany at the State Police, you can give them hard facts. You can ask them for the data. They know how to send it to us. And then we're happy to screen it for you. And then you can say, I have a match statistic here of 1 billion, and what I've gotten so far is inconclusive. Could you please run it? That might be of interest to some people. I don't know. But in the interim, we are working on some cases where trials are coming up and you can't wait. But that's not my goal here today. It's you have this amazing system in Albany. It will solve cases. It will help you find the right people, maybe find out, maybe you have the wrong person, but more often it will help you identify and convict the right people, get them off the street, and create a safer society. Thank you.